Hello, I'm Dr. Israel Barken, the Medical Director of the Prostate Cancer Research and Education Foundation. Today I'm going to discuss a special topic, the circulating tumor cell. But to give you the overall context, I would like first to mention a book that I read recently. The name of the book is The Creative Destruction of Medicine: How the Digital Revolution Will Create Better Healthcare by Dr. Eric Topol. This digital revolution enabled us to use technology to bring medicine from population medicine to individualized medicine, and this is exemplified by the technology related to circulating tumor cells. So let's go now and discuss the topic of circulating tumor cells. Circulating tumor cells, like the name implies, are tumor cells that are floating in the bloodstream, in the circulation. These cells are shedding from the primary tumor or other location and are floating in the circulation, and capturing them may give us information about the tumor. So let's talk first about the circulating tumor cells, which this technology existed for a while, and the company doing this test is named Cell Search. And this was done usually on patients with very advanced disease before taking chemotherapy or not. Baseline circulating tumor cells had prognostic value in metastatic castration-resistant prostate cancer, and any changes in the number were related to prognosis. So prior to the treatment, if there was a high number of circulating tumor cells in the blood, the prognosis was not so good. And on the other hand, if the treatment helped, it would manifest itself by dropping the circulating tumor cells. So for the time being, there is not a clinical utility to try to make decision of treatment based on the circulating tumor cells. And you are aware that what we are doing now, we are looking only at the number of circulating tumor cells. And now there is a new technology that was revealed very recently, which is very, very significant. Recently, there was a paper published that revealed that there is a new technology regarding circulating tumor cell. Please see what this title says. High purity prostate circulating tumor cell. High purity means that they were able to isolate only prostate cancer cell and avoid incorporating perhaps white cells or any other cells. And please see also it says here singular in a tumor cell. A single tumor cell was isolated by a nanotechnology that captured those cells, and by using a special laser microscope that was able to dissect one cell at a time, and one cell at a time was subjected to sequencing. Sequencing is the ability to break down the sequence of the genes on the genetic material, on the genome, trying to tell us specifically that cancer cells, what is the genetic makeup of that specific cancer cell in that specific patient in that specific point in time. So what is it so unique about isolating a single cancer cell? Well, the revolution is that we are able to look at molecular pathology of prostate cancer revealed by next generation sequencing. That means that we are able to take one cell and do a special, rapid, better sequencing of the genetic material, and this gives us opportunity to create a personalized therapy for each individual according to his cancer. Now we have to remember that the way we look at prognosis mainly is to look at the cancer grade, like Gleason, the stage, where is the cancer, look at PSA, look at some tests, but we are lumping all these patients together according to this group information. Now we are talking about ability to be able to isolate a single patient with his single cancer cells to tell us specifically what's going on. Because when you look at Gleason, You get the glycan from a biopsy material that you take one time from a biopsy or one time during surgery. But when you are able to capture cancer cells in the blood, that means we call it liquid biopsy, we are able to get genetic information on the cancer in different points of time. For example, not only that every patient has a different type of cancer, even if it's the same glycan, he has different genetic makeup of his cancer. That genetic makeup is changing over a period of time, even if you don't do any treatment. And when you do treatment, there is also change in the genetic information. So this ability in point of time to take a blood sample and isolate the cancer cells and try to find out prognosis or try to isolate what kind of genetic makeup that patient has, because that may open the door for really personalized treatment. Summary: these novel findings have significantly increased our knowledge of the genetic basis of human prostate cancer and the molecular mechanism responsible for disease progression and treatment resistance. Some of the lesions are potential therapeutic targets. Studies along this direction will eventually make it possible to design personalized management plans for individual patients. This type of checking one single cell by using the Velcro type of nanogram, capturing a single cell from the blood and being able to isolate them and being able to later on study those individual cells, either by genomic evaluation or by other biologic testing on those cells, really opens the door to a brand new world that will personalize and make the treatment much more specific to the patient with prostate cancer. This is done in Cedars-Sinai in Los Angeles. Please stay involved in this technology, because it most probably will have to be generated by patient request to their doctors to try to get the test and try to advance the cause of individualized medicine. And again, I come back to Dr. Eric Topol that wrote the book Creative Destruction of Medicine. This is a different world of being able to do individualized medicine and not lump patients together to statistics and try to peg the patient to part in table, but trying to fit the science to the patient and not the patient to the science of statistics. Stay well, stay informed, and any one of you who buys the book, The Creative Destruction of Medicine, if you don't like it, I'll buy the book from you. I think this is a very important book for all patients to read and understand and see which way medicine is going in the near future. Dr. Eric Topol and myself, both of us, feel that the future is actually now. Stay tuned, stay involved, stay informed, and have fun. Goodbye. If you have any question, please call our foundation at 619-906-4700 or write to us an email at info@pcref.org. Thank you. Goodbye.